Good morning. What has zoology ever done for you is my question. And perhaps we can see in the history of biomedical science that studies of comparative physiology have really allowed us to gain valuable insights into the way in which ourselves and other vertebrates function, particularly in extreme environments. I want to talk to you today to use two model groups of animals, the penguins and the seals, the true seals, the phosid seals. And I think these provide good models for understanding adaptations to these extreme conditions. And the reason for this is that they have both a terrestrial and an aquatic stage of existence. So adaptations to cold in air and are in water are essential for their survival. And not only their survival, but their ability to thrive in these polar environments. The penguins and many other polar animals will survive in some of the most extreme conditions on Earth, where the air temperatures that they're experiencing are well below their core temperature, where you might expect at extreme times for there to be an 80 degrees Celsius temperature gradient between the core and the environment. And they experience wind chill factors while they're out on air. But of course, as soon as animals enter the water, they experience, again, low temperatures, not to the same extreme, but they are faced with the large increase in thermal conductivity of the water, which is 25 times greater than it is in air. So there are great thermal challenges for these groups of animals. And what I'd want to do today is to explain or maybe review some of the adaptations of these groups, the penguins and the seals, and then go on to talk about some of my own research and collaborative research with colleagues, particularly around the terrestrial phase of their life cycle. As we can see from a range of studies across both birds and mammals, there is large geographical variance in the thermal tolerances of animals. So in the bird groups, we see extended thermal neutral zones in higher latitudes, well down close to freezing. In the mammal groups, we often see these lower critical temperatures extending many degrees below freezing. So we see a large variation in the thermal tolerances of groups of animals across the world. And of course, we are one group that sits within that tropical thermal tolerance based on our evolutionary origins. But if we look within the birds, we see a large variation in core temperature, even within individual groups. And there's only really one group of birds, the flightless ratites, that have significantly lower temperatures. All other groups show a large amount of variation. And we see the penguins being in this lower distribution in comparison to the higher body temperatures we see in most birds. And these animals are able to maintain this higher body temperature because of their insulation properties. And if we look at the relationship between insulation and thickness, which I'm sure many people are well aware of in their own clothing, we can see that there's a strong relationship between insulation properties and the thickness. And in the case of feathers, we see the highest thermal conductivities, even better than fur and much greater than fat as an insulated material. And in species such as the emperor penguins, we see up to 20 different types of feathers providing effective insulation for these animals, both in air and also to provide contour feathers to reduce wind chill and to improve their hydrodynamic properties. Even at these lowest temperatures of these animals that are experiencing during the winter, we see the maintenance of core temperature around about 37 degrees. Considerable variation, it does exist, but they are homeothermic endotherms that are maintaining a high body temperature. And this is some really groundbreaking data from a colleague of mine, Caroline Gilbert, who monitored the body temperature of emperor penguins during the winter. And we can see that sustained regulation occurring. But of course, at certain times, these animals are able to conserve this core temperature by changes in their peripheral circulation. And penguins and other polar animals have quite an exquisite control of their peripheral circulation. And so this example of some data collected from the Paul Ponganis lab demonstrates very clearly that the maintenance of the stomach temperature, which is as close to we could get to an internal deep body temperature, is maintained throughout the dive. So the grey shaded area represents the dive, and we can see that core temperature remaining constant. We see temperature within the vena cava remaining at a lower temperature, but still constant. But we see this change in abdominal temperature occurring throughout the dive in a progressive decrease, sacrificing circulation at the periphery for maintenance of the core. And if we look at the terrestrial phase of these animals, we also see that in penguins and in other birds, we see regional heterothermy as a way in which these animals are able to conserve heat, even in quite extreme conditions. So this really nice data set by Eichhorn has shown or measured body temperature from the core, shown in the reds and the yellows, through to progressive peripheral tissues in the green and blue. And what we have here is the relationship between body temperature and time, plotted here in days. And they're able to show that during high periods of rainfall, we see particular large drops in this peripheral temperature to maintain that core temperature. So regional and temporal heterothermy is a very important mechanism by which these animals are able to maintain their body temperature. If we look amongst the mammalian group, again, we see quite a bit of variation in body temperature. And this work comes from Andrew Clark's review paper in Functional Ecology, looking at both bird and mammal core temperature to try and pull apart some of the phylogenetic relationships between body temperature and mammal groups. And again, considerable variation, perhaps more variation than we are familiar with in humans. And we can see here one particular group, the monotremes, which are the egg-laying mammals here, having a significantly lower body temperature, and all other groups showing this wide variation. Where we find the seals, we find them right bang in the middle of a regulated core temperature, somewhere between 36 and 38 degrees Celsius. So again, homeothermic animals, even in these polar environments. The insulation provided by blubber is key to the maintenance of this core temperature. And I don't think this is kind of work we would do now, where we would take a cross-section of a seal and cut it open and look at the thickness of fat. So this work comes from Schmidt-Nielsen's textbook, just demonstrating the thickness of the blubber. But blubber is not an inert tissue of adipose fat. It has blood vessels, a high density of AVAs in to regulate the peripheral circulation in these animals. And again, while diving, we start to see these changes in temperature occurring as the animals make long extended dives. And again, from the Ponganis lab, we see some really amazing measurements of both venous and arterial temperature in diving northern elephant seals. the largest of the phosid seals. And we can see this, again, the dark shows the dive itself. We see this progressive decline in temperature during these dives. And we have dives here, in the case of this example here, of down to 800 meters or so. So we see, often with prolonged periods of diving, this progressive decline in body temperature. So again, that regional heterothermy, that temporal heterothermy, having a number of different functions, both to conserve energy, but also to reduce oxygen demand of the tissues. And finally, just to sort of explore this adaptation during diving, we can see that to extend the dive duration, these animals appear to suppress any of the shivering, any of the thermogenesis that is occurring that we might expect as an adaptation to cold. So work on hooded seals by Fadisham et al. Has clearly shown that by instrumenting the animals and taking measurements of EMG here, we can see this is the period of diving. We see a complete cessation or progressive cessation of shivering. When the animal comes back to the surface, we see an increase in shivering, and then post-dive we start to see large bouts of shivering to try and bring the temperature of the brain and the rectal measurements back up, or the measurements at those sites. So animals have very precise control over temperature regulation. What I want to talk to you now about is some of my own research with colleagues in the University of Glasgow and in Strasbourg and in France, other areas of collaboration, where we're using thermal imaging as a way of non-invasively measuring the metabolic heat production of animals and across a range of species. And already today we've seen at least 2 presentations showing thermal images. We're seeing this technology advancing very rapidly from older liquid nitrogen-cooled devices through to our iPhone devices. And I was lucky enough to get involved in a project to look at thermoregulation in emperor penguins in Dumont d'Urville Station in the French base there, where André Ancel and Caroline Gilbert had deployed a thermal imaging camera on the ice, the first time to take measurements of these animals around the colony. And they took measurements ranging from air at below -20 up to about -10. Periods of quite high wind speed where the wind chill was quite significant for the animals that were being measured. And so what we're able to do is not only get these magnificent images of animals and where they're losing heat from, we're able to identify clearly the sites of heat loss. And so not surprisingly, it's the well-insulated regions that are cooler in darker blues and reds. And the hot areas where heat is only leaking out of are from some areas around the head and the flippers. And so here you can see that blocked out in red here, this is the small proportion of measurements that are actually above 0 degrees Celsius in terms of their surface temperature. We're able to extract these measurements, and what was of interest to us was that we looked at the temperature gradient between the surface and the air, and we're able to see that we had temperature differences here, positive gradients from some of the main areas that were leaking heat, but the regions where the animals were becoming cooler than the surrounding air. And this counterintuitive result to us was really surprising. And what we understand is that this is severe radiative forcing, radiative cooling occurring at the surface, so much so that the energy balance of the surface is below the surrounding air. And I think if we think about this phenomenon a little bit more carefully, we can start to see from our own lives when we maybe park a car on a cold winter's night where we have a clear sky. We'll start to see condensation on the surface of the sky, surface of the roof of the car, and then ice formation on the surface where it's radiating to a clear sky that may be 30 or 40 degrees below air temperature. And this has consequences for the energy balance, we think, for these animals. And the effects of this radiative forcing means that we have a positive temperature gradient for heat to be transferred by convection to the surface of the penguin. It's not often occurs, but it may occur on conditions where we have this severe forcing in these particular types of environments. And we see here that we have heat loss from the feet. We have heat loss from the head and the flippers. And the overall balance, of course, is a heat loss, a loss of metabolic heat production from the animal itself, despite this small convective saving. But of course, the only reason we think that these animals can tolerate this environment is through their behavior. And some work which we haven't done yet is to explore the energy savings around huddling behavior in these animals. But we have done some measurements of animals in the laboratory, in this case on rabbit pups, where in a series of experiments we looked at the total energy expenditure of pups when they were isolated just as single individuals, or when they were living in groups. And if we look at the total energy expenditure, we see that there is an increase of between 20 and 50% in energy expenditure when these animals are cold challenged, when they're isolated. So the behavior of the animals is key to their energy expenditure. And we also see less pronounced changes in body temperature occurring when these animals are living in groups. So we think the same processes are occurring within these emperor penguin huddles, allowing these animals to save energy and to maintain their body temperature. Following on from this work on penguins, it was obvious to us that there were other groups of animals that had this behavioural thermoregulation, this ability to save energy by living in groups. And so I think what's really fascinating for me is this mix of how physiology and behaviour go hand in hand to understand the thermal tolerances of these animals. And again, working with my colleagues in Paris and in Strasbourg, we had a PhD student who investigated this huddling behaviour in the southern elephant seals, this time in Kerguelen Island in the Indian Ocean. And what's fascinating about these animals is that the southern elephant seals, like only one or two other species of seals, go through a catastrophic molt each year. They lose their entire hair, and not only their hair, but they also lose the upper epithelium. And this is an annual molt, and we think this has a major energetic cost for these animals. So there's certain stages of the life cycle that we think these animals are really challenged thermally, and they must come ashore to be able to molt. And this is an opportunity for us to measure their surface temperature, because they need to bring the blood flow up onto the surface of the skin to allow the hair follicle to regrow each year. So a field campaign was undertaken for 4 years, where the actual behavior of the animals were monitored very carefully. And we see a sequence of behaviors as the animals come ashore. they search the area of beach, they aggregate together in these large wallows, these muddy wallows where they share body heat. And what was surprising to us was that at this time, the animals are moving much more than we expected. We expected them to be minimizing their heat loss or minimizing their energy expenditure by remaining relatively stationary. But they moved up to 0.5 kilometre a day on average around the colony, finding these suitable habitats. And what we were able to show, or what our PhD student Laureline Chase was able to demonstrate very clearly, was that we detected changes in the surface temperature of these animals during different stages of the molt sequence. So here you can see some thermal images of an animal midway through its molt, with the cooler area representing the unmolted skin and the warmer skin surrounding it. And you can see here from the graph, looking at the temperature gradient between the body and air, that we see an elevation during this stage. So this is a key stage, we think, when these animals are having to perfuse the skin surface to effectively grow their hair and allow them to return to sea. A number of stomach temperature loggers were also deployed on a smaller sample of animals, and we compared the stomach temperatures between when the animals were aggregated together in a huddle and when they were isolated. Colleagues of mine were the ones to implant these stomach temperature pills. I think that's a really challenging job for the biologists in the field. And what they were able to show was that animals that were isolated had higher, on average, body temperatures or stomach temperatures than those during aggregation. And this again seems to be counterintuitive, perhaps, that these animals that are isolated have a higher body temperature. Why are they not conserving heat when they're isolated? Well, we think one of the reasons is that when they get into the huddles, they may well be able to allow their metabolism to drop and reduce their metabolic rate when they're in the huddle. When they're in the huddle, they've got the potential that they could overheat because of the reduced surface area exposed for heat loss. Through some quite pioneering work of Laureline, she was able to implant heart rate loggers into southern elephant seals for the first time in the field, and successfully was able to look at how the metabolic rate of these animals differed according to how aggregated the animals were, such that we see a negative relationship between metabolic rate and aggregation. The greater time that they spent in the huddle, in the wallows, they had lower metabolic rates, suggesting here we're seeing a very obvious metabolic saving for these animals. I think, again, highlights this importance of group living for these animals in these extreme environments. So I hope I've been able to demonstrate to you that there is real value in looking at some of our polar species, our polar animals that are living in these extreme environments for humans, and not only living but thriving and successfully reproducing in these environments over many years. And I think what this research is showing us is it's very difficult to extract just simple physiological or anatomical adaptations of these animals without considering behavior as key to them. And I think that's a really important contribution I hope that zoologists and animal biologists can make to the field of human physiology and extreme environmental physiology by starting to infuse some ideas around behavior, which I'm sure many of you will also be interested in. Future research that we're involved in is looking at a range of species from small birds, Svalbard ptarmigan with Andreas Nord in the Arctic, and also some really exciting work on thermoregulation in king penguins with Agnes Luden. And finally, some studies that we've just completed on the development of insulation in albatross chicks in South Georgia. So I'd like to thank all the funders and all the people who have been involved in the research over the years. I'm very much reporting a huge team effort from a number of collaborators, and I'm very happy to take any questions that you might have. Thank you.